Okay, so yes, I'm just going to be giving you a brief overview of the Pacific project. So in a roundabout way, this stands for Pathways of Chemicals into Freshwaters and their Ecological Impacts. And I'm just going to start off by giving a brief introduction to the project, the main issues that we're addressing, a description of some of the approaches we're using and our expected outcomes. So this is a 4-year project. The consortium comprises of 3 research institutions and England's environmental regulator, the Environment Agency. I'm going to be leading the project. I'm based at the UK Centre for Ecology and Hydrology in Wallingford. At the University of Bath, we have Barbara Kasprzyk-Borden, who is an environmental chemist who specialises in contaminants in the environment and wastewater epidemiology. At the University of Oxford, we have Michelle Jackson, who is a freshwater ecologist specialising in multi-stressor interactions and their impacts on freshwaters. And Kerry Walsh at the Environment Agency leads their development of DNA-based tools for regulatory monitoring. So at a sort of high level, the Pacific project has this objective to develop a predictive understanding of the risks of micropollutants to the microbial health of freshwater ecosystems. And in the next few slides, I'll just give you a brief sort of overview of the types of micropollutants we're interested in, particularly mechanistically, what types. And then really trying to justify actually this focus, which I think is different from many of the other projects, focusing on the microbial aspect of freshwater ecosystems. So micropollutants in freshwaters. So these are defined as contaminants that are at or below the microgram per litre level. And they are incredibly diverse, so hundreds to thousands of anthropogenic compounds and a sort of ever-increasing list. And many of these make their way into the environment. And they come from a whole range of different sources, so pharmaceuticals, personal care products, biocides, household chemicals, agrochemicals. That's a far from exhaustive list. So, really diverse sources. And many of these can actually make their way into freshwaters through sort of broadly 2 main pathways. Diffuse sources, so typified by things like agrochemical applications, but also sometimes road runoff, runoff of anthropogenic surfaces. These can make their way into freshwaters. And then the other main pathway is point sources, really sort of typified by the wastewater network that releases both treated and occasionally untreated wastewater into freshwaters. And one of the things that we're really interested in, because this is sort of an almost overwhelming diversity of chemicals that make their way into the environment, is that a subset of these chemicals, a significant subset of them, are targeted at microbes. They're designed, manufactured, and applied to kill microbes, and that includes antibiotics, antivirals, fungicides, general biocides. So there's a whole range of chemicals we know by design will target microbial communities. There's also increasing evidence that a wider range of non-antimicrobial chemicals can also impact the growth of microbial communities. In the bottom right here is a paper that came out a couple of years ago that showed about 25% of non-antibiotic pharmaceuticals will impact the growth of microbes within the gut microbiome. So there's this sort of known chemicals which we know will target microbes, and there's potentially a longer list of unknown chemicals as well. And this sort of really leads to this sort of major knowledge gap that we know a diverse array of chemicals make their way into freshwater environments, but what we don't really understand is the impact of these chemicals on freshwater microbial communities. So the next thing is why microbes? Why are we interested in these? And there's a list here, and I think some of these points are more important than others. So, they're the most abundant biological component, numerically, if not in biomass. They're the most taxonomically diverse component, by orders of magnitude, I think, compared to other freshwater biodiversity components. But I think really sort of critically, these have been defined as the biogeochemical engine. So they're responsible for organic matter turnover, cycling of carbon, nitrogen, phosphorus. They control river metabolism, this sort of balance between the utilization and production of oxygen in freshwaters that has wider impacts on freshwater ecosystem health. And they control greenhouse gas fluxes as well. So they're the sort of engine that's controlling a whole load of these processes that are important for wider freshwater ecosystem health. They can also be directly responsible for the health of organisms in freshwaters as pathogens. And as I've already said, they're sensitive to a range of chemicals, anthropogenically produced chemicals that are designed to target these organisms. So these antibiotics, biocides, and so on. So how do we actually go about measuring microbes? And in some ways, this has parallels to the sort of conventional biomonitoring approaches. We can think about shifts in community composition, so the taxonomic responses upon exposure to a chemical. So in a very simple example, we have a healthy microbial community in a freshwater. It's exposed to a chemical, maybe an antibiotic, and this causes a change in the community composition. So specific taxa may get up or down regulated depending on their resistance or sensitivity to chemicals. And this gives us the ability to potentially identify new bioindicator taxa that are tailored to specific chemicals. But there's also this functional aspect as well, these processes that microbes are doing. So we can count sort of high level functional processes such as cell density, cell activity. We can probe the biogeochemical processes that these organisms are performing. We can look at things like enzyme activity, gas fluxes, and we can look at these functional genes, the genetic repertoire that's involved in controlling these processes and the markers for the community composition. And we have a range of these sort of off-the-shelf methods in my laboratory that we can apply for this. So high throughput sequencing that will probe both the taxonomic composition of these communities and the functional responses. We can use a range of high throughput assays to characterize these different functional endpoints such as cell density, physiology, these biogeochemical processes, particularly enzyme activity. Okay, so this slide gives a sort of high-level overview of the data generation work packages within this project. And what I've done is put these across a gradient along the top. On the left-hand side, we have our most tractable, controllable, reproducible experimental data generation activities. And on the right, we have the highest sort of relevance, but also the highest complexity. And this is sort of how we broadly structured our data generation to link between these different phases. So I'll just briefly go over these and give you a sort of very high level view of what we're planning to do. So as I've already mentioned, there's this sort of known knowns in terms of chemicals that are likely to impact microbial communities, but there's also these unknown potential impacts because microbes are not normally screened as part of ecotoxicological assays in terms of understanding, bringing new chemicals to market, or at least it's done against a very, very limited repertoire of microbes. So our intention is to take advantage of the fact that microbes are very small. When we run a microcosm, we can really miniaturize these. So these 96 well plates, each of them contain 100 micrograms of sediment, which is about 100 million individual bacterial cells. So we're still looking at complex microbial communities, but just the whole thing is miniaturized. And because of that, we can screen a lot of chemicals. So the intention is for this project to screen about 1,600 different pharmaceutical chemicals, metals, biocides, agrochemicals, household chemicals, to do a sort of chemical prioritization exercise. So understanding which chemical types and chemical classes are likely to impact microbial communities, and then layer this on top of actually looking at what chemicals are likely to make their way into the environment. The next part of work package 2 is to perform much more detailed analyses. So this will be looking at concentration ranges, looking at a range of these different endpoints, so functional endpoints, metabolic endpoints, taxonomic endpoints, to look at generating new thresholds. So these would be against what concentration are we likely to see these different types of responses from the community composition to these functional endpoints. Moving sort of to more complex, more realistic experimentation, we're working with the University of Oxford. We're going to use this extreme mesocosm system. This is in work package 3. These are larger mesocosms, so about 25 centimeters across in the scale of a microbe, gigantic. These will have much more complex exposures, looking at real world exposures of wastewater with a number of different multi-stress interactions layered on top. And then finally, the most complex, but with the highest level of relevance, is the field sampling. So we intend to sample across 2 different river catchments, the Bristol Avon and the River Thames. And this is really where we pull all of our different techniques together. We can look at chemical composition using high resolution mass spectrometry. We can look at these taxonomic endpoints. We can look at these functional endpoints to understand whether or not the the more sort of simplistic experimentation we can do in the earlier work packages will then, those endpoints, we can actually identify them in much more complex systems. And like many of the other projects, we intend to sort of link the, to scale up using modeling-based approaches. So obviously sampling in the field has its limitation. Using models, we can scale up both spatially across whole river catchments and also temporally to look forward as to how things may change. And we're using two sort of linked modeling approaches. So one is a point source modeling approach. This is a cube model, a hydrological model that's widely used by UK environmental regulators. And this will be further developed into a water quality extension based on a previous modelling experience we have at UKCEH. So this will give us predicted environmental concentrations of chemicals across river catchments from wastewater. For the diffuse component, we're using a model called INVEST. This takes advantage of the UKCEH land cover map and crop map to look at predicted agrochemical applications to land and use hydrological routing to predict loads reaching freshwaters. And the intention is to combine these, and one of the sort of key outputs of this modelling exercise is to produce these catchment risk maps, so scaled up risk maps that look at concentrations of exemplar chemicals, but then linking with our other components actually start to put on some of these experimental thresholds around shifts in taxonomic composition of communities, importantly shifts in these functional endpoints, these microbial functional endpoints, so we can actually map and look at where the microbial integrity of freshwater ecosystem health is potentially under threat at the catchment scale. So a key component of this is obviously trying to work with stakeholders to actually develop some of these sort of outputs of this project into useful tools. So as I said, the risk maps is something we'll be working closely with our partners, the Environment Agency. The sort of risk mapping is a language that I think is quite widely used within regulatory monitoring, and I think these tools will be useful to be able to target further monitoring. Another major output is actually because of the sort of new way in which we're thinking about looking at the freshwater microbial communities. The intention is we'll have a suite of novel bioindicators that can look at the taxonomic shifts of microbial communities, these functional shifts, these genetic shifts, as ways of diagnosing the natural environment in terms of understanding the ecosystem health and particularly understanding the types of multi-stressor impacts that are affecting these communities. The other component is this sort of forward-looking, and this is similar to some of the other projects, I think, really, where we can look at scenarios, socioeconomic scenarios, hydrological scenarios, land use scenarios, to look at not necessarily an accurate prediction of what will happen, but more what could happen under these different scenarios in terms of, say, changed rainfall or changed land use within the catchment. The project will be working closely with a range of different stakeholders. We have a project advisory group comprising of water companies, rivers trusts, and to really sort of give us advice throughout the project, particularly around designing our sampling campaign and sort of linking it with existing data sets. And I think that's everything from me, so I'll be very happy to answer any questions.